This is the study of Apis mellifera and the beneficial biological activities of propolis on the human liver. The interactions of Apis mellifera and humans on a biological level. What are the beneficial biological activities Apis mellifera products, such as propolis, have on the human liver? The benefits of the biological materials produced by Apis mellifera are numerous, and can prevent or aid in many human neurodegenerative disorders and in general maintenance of health. The benefits focused on will be about the hepatoprotective properties. Hepatoprotective is the ability to protect the liver by a chemical substance via restoring liver functions to normal levels. Hepatoprotective agents are crucial in protecting the liver from damage. The liver is a major metabolic organ and gland in the body. It is also important for detoxification and immune function. Propolis is a bee product that consists mainly of resin, beeswax, essential oils, pollen, and other organic compounds. Apis mellifera produces both honey and propolis. Here you can see all the beneficial biological materials produced by honey and propolis. For this research, we will be focusing on the hepatoprotective properties of propolis. Hepatoprotective properties of propolis have been investigated against mercury in 2,3,7,8 tetrachlorodibenzo P-dioxin, which causes skeletal deformities and kidney defects, as well as weakened immune responses due to its interactions with the liver. One study done by Wali Avula, Ali Khan, Mushtaq Rahman, Akbar, and Masudi have discussed how KPET, material collection preparations of propolis ethanolic extract, showed protective effects against INH RAF induced hepatocellular damage by inhibiting oxidative stress, maintaining balance of antioxidant enzymatic and non-enzymatic, and distinct decline in COX-2 and NF-kB expression in rodents. The presence of flavonoids and phenolic acids is responsible for hepatoprotective activity by decreasing the level of hepatic markers, lipid peroxidation, and attenuates free radical scavenging. Therefore, treatment regimen executed with groups 3 and 4 demonstrated remarkable hepatoprotective activity by bringing the enzyme levels and other biochemical parameters towards normal as compared with group 2. Group 3 showed maximum recovery of hepocytes, specifying its specific hepatoprotective activity. Similarly, Group 5 showed marked recovery, but recovery was lesser compared to Group 3 and more significant compared to Group 4. In this study done by Yangi, Kinskis, Ustener, Denser, Osbeyer, Tenkin, Ustener, Kolak, and Intok aimed to explore the potential in vivo anti-inflammatory, antioxidant, and anti-apoptotic properties of propolis extract on lipopolysaccharides. LPS-induced inflammation in rats. Anti-apoptotic is something that prevents apoptosis, a type of cell death that occurs when a series of molecular steps within a cell leads to its death. From this experiment, all the test groups that were on propolis found their inflammation to be significantly lower. The study pointed out the protective effects of propolis extract in LPS-induced inflammation and oxidative stress and showed that those with higher consumption of propolis had higher antioxidant and anti-inflammatory activities. The data of the study indicates that the propolis could serve as a potential approach for treating inflammatory liver diseases through reducing inflammation and free oxygen radical production. Tutunchi, Arif Hosseini, Abrahimi, Magahani, did a study in 2022 about propolis supplementation on metabolic parameters in liver function obese patients with non-alcoholic fatty liver disease, or NAFLD. The greatest reduction in serum ALT was observed in propolis group, while control group showed the greatest change in AST and AST/ALT ratio. Alanine aminotransferase, an enzyme found in the liver that helps convert food into energy. ALT levels in the blood are usually low, but when liver cells are damaged, ALT is released into the bloodstream. High levels of ALT in the blood can indicate liver damage, cancer, or other diseases. Propolis supplementation has been shown to influence glucose metabolism through increasing glycolysis, glucose uptake from peripheral tissue, and glucose utilization in the liver, reducing carbohydrate absorption in the intestine, inhibiting glucose from reaching the liver. Propolis and MI supplementation on liver function in the study indicates reduction in the serum ALT by propolis supplementation and produces a favorable effect on the serum AST and AST/ALT ratio in the control group. In the final study done by Guimarães, Melo, Pavia, Bueno, Beretta, Torquato, Nantes, and Rodriguez investigated the protective effects of the glycolic extract of B. dracunculifolia, or GEBD, against oxidative stress in isolated rat liver mitochondria. GEBD inhibited the formation of the intermediate and final products of lipid peroxidation during the oxidative chain reactions. Besides the lipid protective effect, GEBD also prevented the oxidation of mitochondrial proteins induced by iron or ferrous ion. A. ROS generation was assayed by DFC fluorescence. B. Induce lipid oxidation of mitochondrial membrane by GEBD estimated T bars. C. Induce thiol oxidation of mitochondrial proteins by GEBD measured with DNB. D. Induce GSH oxidation by GEBD estimated fluorometrically. The antioxidant activity exhibited by GEBD in non-biological systems was confirmed also in mitochondria, preventing the oxidative damage of lipids and proteins. Through the collected research, propolis and its compounds are shown to be hepatoprotective in nature. This is important in maintaining a healthy liver, which is part of the human immune system and detoxification system. These research studies have shown propolis to be important in aiding diseases such as non-alcoholic fatty liver disease. The anti-inflammatory, antioxidant, antibacterial, antifungal, and anti-apoptotic properties of propolis is highly beneficial in protecting the liver. Propolis influences glucose metabolism through increasing glycolysis, protecting the liver from oxidative damage. The significance of these research shows the benefits from Apis mellifera biological products such as propolis. While not endangered or in need of conservation, research on propolis highlights the importance of honeybees. Not only do they pollinate 1/3 of human crops, but can protect against degenerative diseases and maintain human health.